Hi, grade 9. Are you fine? Today we are going to practice about the digestion. Let's begin with the digestion of maltose by Maltese. Maltase is an intestinal enzyme that acts on maltose, which is the malt sugar, and permits its digestion. To verify this hypothesis, we perform an in vitro digestion of maltose. The following are the steps of the experiment. Put 5 milliliters of maltose solution into two test tubes A and B. Add small amounts of maltase into tube A only. Place the two tubes in a water bath at 37 degrees for 30 minutes. Look at the steps of the experiment. If they ask to state the steps, you will use them or the given as steps 1, 2, 3. If they ask to describe the experiment, we use connective words and we make them as a scientific text. Okay. After 30 minutes, we measure the quantity of maltose in each of the two tubes A and B. The measured results are reviewed in the table below. Time in minutes, it starts with 0 to reach 30 minutes. Quantity of maltose in tube A, look at the quantity. It starts with 5 and it ends with 0. Quantity of maltose in tube B, it's still 5. Define the term in vitro digestion. In vitro digestion is the breaking down of complex food molecules into simple ones outside a living organism that is in a test tube, which is made in laboratory. Specify the common conditions and the variable condition of the above experiment. For this experiment, the common conditions are 5 milliliters of maltose, temperature 37 degrees, and time 30 minutes, since they are common for tubes A and B. While variable conditions, this is one condition, variable condition is enzyme, which is maltase, since it is present in tube A only. Give the name of the test tube B. The test tube B is a control tube. We use the control tube in order to compare the result. Explain why the test tubes are placed in water bath at 37 degrees. Actually, this is the temperature of body temperature. The test tubes are placed in water bath at 37 degrees since it is the favorable temperature for the optimal activity of the enzyme. Why is it the favorable temperature? Because it's the body temperature. Inside the body or in vivo, the digestion occurs at 37 degrees, which is the most available temperature. Pick out from the text the tested hypothesis. Maltase is an intestinal enzyme that acts on maltose, which is malt sugar, and permits its digestion. Draw on the same graph the curves which shows the variation in the quantity of maltose in the two tubes A and B in function of time. In function of time means that on x-axis we use the time, we choose the appropriate scale, and then on y-axis we put the data of each tube A and B, for example, using two colors, and finally we connect them and to obtain the curve, two curves. Is the hypothesis validated? Justify the answer based on the analysis of the results of the above table. Yes, the hypothesis is validated because when Maltese because when Maltese is present, it transforms maltose into another type. We notice that the amount of maltose decreases when Maltese is present. Yes, the hypothesis is validated. In tube A, where Maltese is present, the quantity of maltose was 5 milliliters at the beginning of the experiment, and it decreases progressively to reach 0 milliliter after 30 minutes, whereas in tube B, where maltase is absent, the quantity of maltose remained constant, 5 milliliters during 30 minutes. Maltose is also known as malt sugar, is a disaccharide formed from 2 units of glucose. It is produced when amylase breaks down starch. It is found in germinating seeds such as barley. As they break down their stored starch to use for food. It is also produced when glucose is caramelized. Caramelization is the browning of sugar, a process used extensively in cooking for the resulting specific temperature, where the series of the reactions of caramelization starts. Refer to the text above. Pick out from the text a polysaccharide, the enzyme digesting it, and the product produced. Polysaccharide is starch, enzyme is amylase, and the product is maltose. Indicate the two ways by which maltose is produced. Maltose is the disaccharide produced when amylase breaks down starch. It is also produced when glucose is caramelized. The table below gives the caramelization temperature for 5 different sugars. The adjacent figure shows creamy caramel sugars. Fructose, which is 110, galactose 160, the same for glucose and sucrose, and 180 for maltose. This is the caramelization temperature. And this is the caramel. Refer to the table. Classify the sugars in the table into monosaccharides and disaccharides. Monosaccharides are fructose, glucose, and galactose, while disaccharides are sucrose and maltose. Choose from the sugars of the table the one that gives negative result with Fehling solution, which is the sucrose. Translate into the information into a histogram paragraph. We use the data on x-axis and numbers on y-axis. According to your acquired knowledge, name the organ of the digestive tract where digestion of sucrose occurs, given the suitable pH for the occurrence of this digestion. The organ is the small intestine and the pH is 8. This is the end of our video. Thank you for watching and see you in the next one. Goodbye.